So by these 20 to 30 efferent ductules, these efferent ductules actually it attaches the it connected the upper pole of the testis with the head of the epididymis. Then here the each efferent ductules become highly coiled and it forms the head of the epididymis, and then these tubules they end in a single duct and which traverses through the body and tail of the epididymis. Then when this duct or tubule it exceeds the epididymis, it is named as or it is continued as ductus deferens or vas deferens. Let's say it so it will be clear. So look at that. We told you that each lobule contains seminiferous tubules 1 to 3 and some interstitial cells of the lady. So these are the seminiferous tubule and it contains some convoluted portion in front and some straight portion behind. And these from all the seminiferous tubules, when they connected to each other, they forms a plexiforms of network. This is known as the rete testis. And from the rete testis, it gives rise to about 12 to 13 efferent ductules. And they are highly coiled in the head of the epididymis, and the body and the tail they are continued as a single ductule. And when it exits the epididymis, its name is then ductus deferens or vas deferens. Now, where is the interstitial cells of the lady? The interstitial cells of the lady they are present in between seminiferous tubules. So this is the seminiferous tubule, cross section of a seminiferous tubule. This is another seminiferous tubule. Here will be another seminiferous tubule, another seminiferous tubule. So look at that, between adjacent seminiferous tubule there are some cluster of cells. This is the Leydig cell. This is another Leydig cell. This is another Leydig cell. So the interstitial cells of the Leydig they presents between the Sertoli cells. Now come to the microscopic structures. Now we will see the microscopic structure, microscopic formation of the seminiferous tubule. So seminiferous tubule it contains actually two distinct type of cell. One is the spermatogenic cell and another one is the cells of Sertoli or Sertoli cell. The spermatogenic cells it includes in the outer zone or the basal zone spermatogonia, in the intermediate zone primary and secondary spermatocytes, and in the inner zone there is the spermatid and spermatozoas, and their function is the spermatogenesis. Then come to the cells of Sertoli or Sertoli cells. These are the elongated polyhedral cells and they extend from the basal membrane up to the lumen of the seminiferous tubule, and they have several function, multiple function. We will learn one by one. Before going to the details to the cells of Sertoli, we will see which one is seminiferous, which one is spermatogenic cells, and which one are the Sertoli cells. So we are seeing the microscopic structure of the seminiferous tubule. This is a cross section under microscopy of the seminiferous tubule, and so this is the basement membrane or basal zone, and this is the lumen of the seminiferous tubule. And you can see in the basal zone or the outer zone there are some cells. These are known as the spermatogonia. Then in the intermediate zone there are primary spermatocyte and secondary spermatocytes. These are the spermatocytes. And in the inner layer there are some spermatid and spermatozoa. So in the inner layer there is spermatid These are the spermatozoa, which is located in the lumen of the seminiferous tubule. So these are the spermatogenic cells. So these spermatogonia, spermatocytes, spermatids, and the spermatozoa, all these cells are spermatogenic cells, and their ultimate goal is to produce, and the ultimate goal is the formation of the spermatozoa. And then which one is the Sertoli cell? Look at that. This elongated polyhedral cell. This elongated polyhedral cells is known as the Sertoli cells, and they have a nucleus, and they are extended from the basement membrane up to the lumen. So they are huge. And look at that. Two adjacent Sertoli cells are connected to each other. Two adjacent Sertoli cells are connected to each other by a tight junction, and this tight junction ultimately form the blood testis barrier. So what is this? The blood testis barrier. It is a barrier between the testis and the blood. So look at that. These are the some tight junction, and these are the blood vessel. So it is a blood testis barrier. It is a barrier or partition which separates the blood from the spermatozoas. Now the question is, why should be there a partition? Okay, at first learn about the blood testis barrier. So we saw that blood testis barrier, these are nothing but the lateral surface of the adjacent Sertoli cells are bound together by some tight junctions at the level of the spermatogonia. These tight junctions mainly forms the blood testis barrier. So what is the function? Due to the presence of this barrier, the circulating plasma protein doesn't expose with the sperm protein. So what will be the consequence if these come contact with each other? There will be production of the anti-spermic antibody, and this will destroy the spermatozoa, and there will be suppression of the spermatogenesis, and ultimately it may lead to the infertility. So why, the question may become, why there will be production of anti-spermic antibody? Because the sperm is naturally present in our body. The answer is, in the fetal life or in the embryonic life, whatever is present in our body, it is considered as self or safe. But anything that is acquired after the birth, is considered as foreign. For example, in male, the spermatozoa is produced, that means spermatogenesis is produced at the puberty age. So before puberty, that means in the embryonic period, there is no spermatogenesis. So as it is produced after puberty, all the sperms or sperm proteins, they are considered as foreign body by our blood cells, which are mainly defensive in function. So they consider the sperm protein as a foreign body, and when they are exposed, when the sperm proteins are exposed to the blood cells, they recognize as foreign body and production of the antibody, and ultimately they destroy it. So that's why to separate them in a separate compartment, there is a barrier which is known as the blood testis barrier. Now we are talking about the Sertoli cells. So what is the function of the Sertoli cells? One of the functions we already discussed, that is the establishment of the blood testis barrier. Number 2, it support, protect and give nutrition to the spermatogenic cells. We saw that between spermatogenic cells, the Sertoli cells lies. So it will definitely give a support, it will definitely give some nutrition and protection to the spermatogenic cells. And the most importantly, it gives it produces some androgen binding protein, some inhibin, anti-Mullerian hormone, and so on. So what is the function of this hormones. Androgen binding protein or ABP. It binds with the testosterone and increases the concentration and the half-life of the testosterone. It helps in the spermiogenesis, and it also releases inhibin. Inhibin, it gives negative feedback mechanism to suppress to the release of the FSH or follicular stimulating hormone, and anti-Mullerian hormone. In embryonic life, in case of male, it prevents the differentiation of the female internal sex organs in male fetus. As we know from the Mullerian duct, there is development of the internal female genitalia, that means uterus, fallopian tube, vagina, etc. So anti-Mullerian hormone, that means a hormone that acts against the Mullerian duct. So there will be no Mullerian duct, no formation of the uterus and vagina in the male. And Sertoli cells, it has some special property which is resistant to the infection, malnutrition and X-ray radiation. And it causes some amount of phagocytosis. So actually, if we talk about the spermatogenesis and production of the testosterone, this is mainly controlled by a axis which is known as hypothalamo pituitary gonadal axis. Look at that. From the hypothalamus, the gonadotropin releasing hormone is GnRH, gonadotropin releasing hormone is released. Then it acts on the anteropituitary and releases FSH and LH, and this LH acts on the FSH and LH acts on the gonad, that is testis. LH acts on the Leydig cell to produce testosterone, and FSH stimulates the Sertoli cells to produce the spermatozoa, that means it causes spermatogenesis. So this hypothalamo pituitary gonadal axis, this is known as hypothalamo pituitary gonadal axis. And when the FSH produces huge amount of the spermatozoa, then when the FSH stimulates the Sertoli cells and produce sufficient amount of the spermatozoa, these Sertoli cells then release a hormone that is known as inhibin. This inhibin, it sends some negative feedback mechanism so that there will be reduced FSH secretion, reduced stimulation of the Sertoli cells, and ultimately there will be reduced spermatogenesis. So this inhibin acts via negative feedback mechanism to suppress the FSH and ultimately to suppress the spermatogenesis when the spermatogenesis is enough. Now come to the interstitial cells of the Leydig. We are talking about the microscopic structure. Now come to the interstitial cells of the Leydig or Leydig cell. So we saw that this is found as clusters in between the seminiferous tubules, and the function is the main important function of the Leydig cells. You also already saw that by the hypothalamo pituitary gonadal axis, it acts. It is stimulated by the luteinizing hormone. It is stimulated by the luteinizing hormone. Acts on the it is stimulated by the luteinizing hormone and acts on the Leydig cell, and this Leydig cell ultimately form the testosterone hormone. And this testosterone hormone is the main hormone for the secondary sexual characteristics of the male pattern. Now, if someone asks about the function of the testis, you will say the function of the spermatogenic cells, function of the spermatogenic cells, function of the Sertoli cells, and function of the Leydig cells. Or as a whole, in short, we can say the testosterone, the main function of the testis is number 1, production of the testosterone, and number 2, that is the production of sperm or spermatogenesis. Now come to the artery supply of the testis. The testis is supplied by the testicular artery, which is a branch of the abdominal aorta. And in case of ovary, the name of this artery is changed into ovarian artery. And this testicular artery and ovarian artery as a whole, their common name is the gonadal artery, as they are supplying the gonad. So the gonadal artery, that means in case of male, which is known as testicular artery, and in case of female, which is known as ovarian artery. These are the branch of the abdominal aorta. And partly they are supplied by the artery to the vas deferens, and sometimes it is supplied by the cremasteric artery. So this is the testicular artery, right and left. So look at that, they are coming from the lateral to the abdominal aorta. So they are the lateral branches of the abdominal aorta. Now come to the venous drainage. The most important thing is the venous drainage of the testis. So the venous drainage, from the testis, about 15 to 20 veins appear from the posterior border of the testis and epididymis, and they unite to form a plexus, which is known as pampiniform plexus. And at the superficial inguinal ring, these plexus unite to form only 4 veins, and at the deep inguinal ring, they further unite to form only 2 veins. And finally, we can find a single vein that is formed in the posterior abdominal wall. And ultimately, this single testicular vein, ultimately on the right side, that means right testicular vein, directly drains into the inferior vena cava at an acute angle, and in the left side, that means left testicular vein, that drains into at first into the left renal vein, and then left renal vein is drained into the inferior vena cava. And this venous drainage is via a right angle, that means 90 degree. So let's see the picture. So look at that. So from the posterior pole of the testis, there is several veins, 15 to 20 veins arises, which is known as pampiniform plexus. And at the superficial inguinal ring, this is the pampiniform plexus. And at the superficial inguinal ring, there are number of 4, and at the deep inguinal ring, there only forms the 2 vein, and at the posterior abdominal wall, they unite to form only 1 single testicular vein. And look at that, on the right side, it directly drains into the inferior vena cava, and on the left side, the left testicular vein, at first it opens into the left renal vein and then into the inferior vena cava. And look at that, the drainage of the vein. The right testicular vein, drain into the inferior vena cava at an acute angle, but the left testicular vein drain into the left renal vein at an right angle, that means 90 degree. So the left testicular vein have to work against a great pressure, and they have to work a lot. So this is correlates with a clinical anatomy that is known as varicocele. It is very common in the testis. So what is varicocele? It is a clinical condition in which the vein of the pampiniform plexus become dilated, tortuous, and elongated, and it commonly affects the young adults. And the varicocele is most common on the left. Why? Because the left testicular vein drains at a right angle in the left renal vein, so the venous pressure is high. in the left testicular vein. This is the number 1 cause. Number 2, the left testicular vein may be compressed by the loaded constipated sigmoid colon. We know in the left side there is a sigmoid colon. So if a patient is suffering from the constipation, the stool material, it may compress the left testicular vein, and there may be hampered of the venous drainage, and there may be production of varicocele. And as well as the blockage of the entry of the left testicular vein in the left side, it may be done by the left renal malignancy. If any malignancy Any tumor of the left kidney, it may compress the left testicular vein and may contribute to the production of the varicocele. Now come to the nerve supply. The testis is predominantly supplied by the sympathetic nerve, that is the thoracic tail to the thoracic element segment of the spinal cord. And last of all, the development of the testis. The testis develop from the testis as well as the ovary. They develop from the genital or gonadal ridge of the intermediate mesoderm. Now come to the descent of this testis. Actually, the testis in the embryological period, they were in the abdomen. The testis usually in the fetal life, they were in the lumbar region of the posterior abdominal wall. That means they were in the abdomen. But later, they descend through the pelvis and the inguinal canal during the 7th and 8th month of the fetal life, and then ultimately goes to the scrotum. So how they are descend? How they descend? The number one, the descending factor for the testis, the number one is the gubernaculum and the processus vaginalis. And as the gubernaculum shortens, they pull the testis along a pouch of peritoneum, which is known as processus vaginalis. Let's see. So that embryological constituent, which is known as gubernaculum testis. Look at that. It attaches the lower pole of the testis with the base of the scrotum, and when the baby grows up in the uterus, these gubernaculum testis, they shorten in length. When they shorten in length, these gubernaculum testis pull the lower pole of the testis to the base of the scrotum. And ultimately, just before birth, the scrotum is in its, the testis is within the scrotum. And as well as beside this gubernaculum testis, another factor plays a important role, that is the some pouches of the peritoneum, which is known as the processus vaginalis. So some pouches of the peritoneal pouch, which is known as the processus vaginalis. It acts as it guides the testis. It guides the testis to descend in the right pathway. So we are talking about the descent of the testis. Now the question may come, when the descent, when the timing of the descent. The descent of the testis begins at the 2nd month of the intrauterine life. Descent of the testis begins at the 2nd month of the intrauterine life, and it reaches the iliac fossa by the 4th month, and at the 7th month it reaches the deep inguinal ring, and it traverses through the inguinal canal during the 8th month, and finally it reaches the scrotum at the time of the birth. So just soon after birth, we should check the testis, either it is present within the scrotum or not. And the gubernaculum testis and processus vaginalis, which is the main factor for the descent of the testis. So just after the descent, so after descending the testis, after keeping the testis in its within the scrotum, the gubernaculum testis usually disappears, and it persists as a rudimentary scrotal ligament, and the processus vaginalis, the proximal part, it dilates, but the distal part persists as a closed vaginal sac. Now, what are the factors that is responsible for the descent of the testis? We already show you that the gubernaculum testis and the intra-abdominal pressure, intra-abdominal temperature. That is one of the most important factors. Why? Because we know that for the purpose of spermatogenesis, we need a lower temperature. But in the abdomen, the temperature is about 37 degrees Celsius, 37 degrees centigrade. And but in the scrotum, the temperature is about 4 degrees centigrade lower than the abdomen. So that's why the scrotum provides a lower temperature, and for the purpose of spermatogenesis in a lower temperature, the testis descends. And there are internal oblique muscles, some arch fiber of the internal oblique. It causes contraction and squeezes the testis, uncurling of the fetal curves, and as well as the some secretion from the testis itself. So these are the factors which causes descent of the testis. Now we will learn about the clinical conditions, that is cryptorchism. What is cryptorchism? It means the imperfect descent of the testis. The testis is not descended completely. It can be divided into another incomplete descent and mal-descent. Incomplete descent, that means the testis is traveling through the normal pathway, but it is arrested in somewhere. That means it may be arrested within the inguinal canal, or in the superficial inguinal ring, or in the deep inguinal ring, or high up in the scrotum. That means they are in the normal anatomical pathway, but they didn't complete the full pathway. They are arrested somewhere. So this is known as incomplete descent. Mal-descent, that means they are arrested elsewhere other than the normal pathway. That means they may be arrested in the inguinal ligament, or in front of the symphysis pubis, or as well as in the perineum, or in the thigh. Okay, so that means they are arrested in the abnormal sites, abnormal pathway. So these are known as the mal-descent of the testis.